morning, class. Today we're going to learn about cell cycle.
Yay, this class is so fun. I don't even understand any of this.
So the cell cycle is how long it takes for one cell to become two.
What? I don't get it.
Oh my lord, today's begun again. I was saying the cell becomes two, that's literally all I said. So mitosis is, oh my god, I need the BioBrainies.
BioBrainies to the rescue! We're here to help you understand cell cycle and more, and we're also going to get rid of that boring teacher.
Welcome to today's BioBrainies podcast about the cell cycle. Yay! Oh, wait, let me fix these glasses first. Oh, whoop. There we go. Okay, so in this video, we're going to specifically talk about the cell cycle and the different phases that are in it. So interphase, mitosis, cytokinesis, and sometimes meiosis, and how that cell cycle is very highly regulated so that there's internal and external checkpoints along the way. And if there is a disruption on that cell cycle, how that can lead to cancer. So let's get started. Okay, so the cell cycle really is correlated with the word cell division. And what happens in cell division is when the parental cell divides into two daughter cells. And so there's the different phases, like I said, that's interphase, mitosis, and cytokinesis. So if you sort of see on this diagram, this blue arrow takes up the majority of this overall time period that the cell cycle takes. And so you can infer that the interphase will take the longest out of the three different phases. And in that interphase, there are three subcategories, and there's G1 and G2. You can sort of think of the G as grow. So in the interphase, usually the cell is getting ready to divide, and so the cell itself has to grow. And specifically in the S phase right here, you will experience this DNA replication, and this process is necessary in order for the daughter cells to result in the identical number of have the identical number of chromosomes as the parental cell did. So that's basically what interphase is. The cell is just getting ready to divide, and the DNA replication is occurring there. The next phase is going to be mitosis. This is going to take the second longest. There's going to be again subphases such as prophase, metaphase, anaphase, and that's basically where the internal parts of the cell are sort of moving around, shifting around, splitting a little bit apart for the cell to actually get ready to divide. But most specifically in mitosis, this telophase part right here is very important because telophase is where the nucleus is beginning to divide. So mitosis is starting to get into the actual division of the cell. And specifically, it's going to be the nucleus that's dividing. So it's also known, this process is also known as the nuclear division, also the karyokinesis right here. And so, yeah. This is a more specific diagram that goes into what exactly is happening. Right here, you can sort of see going from anaphase to telophase. There is the division of this outer layer of the nucleus beginning to divide. But one thing to note is that in order for the cell to fully divide into two, it's very important for not only the outside edges to split, but also everything inside to also split along with it. So mitosis and the cytokinesis, which is going to be the next process, are interdependent. So in mitosis, the nucleus is dividing, but in cytokinesis, the prefix is cyto, you correlate it with cytoplasm. And so in cytokinesis, the cytoplasm, or the jelly-like substance inside the cell, which is this pink area right here in this diagram, that is going to divide in this specific phase. And so as you can see, sort of right here, that's the nucleus that's dividing. Right here is going to be the cytoplasm that's dividing. And after all of these occur, you result in these two identical diploid daughter cells. And so that's basically the result of cell cycle. But there's also one extra step that I could go, and this is going to be meiosis. And meiosis is where the two diploid daughter cells will split once more to result in four haploid daughter cells with only half of the chromosomes that the parental cell had. And so you can sort of see this cell division, cell cycle in your body, usually when you get a cut on your hand, and so cell division will occur to help heal it. Also, it's sort of what's occurring specifically to the embryo of a baby in the mom's body. and how that embryo becomes a baby it is when it is born, and cell division is occurring to form different organs and stuff like that. So, yep, that's basically my part, and here's Heaji to come and push me over.
Okay, now that she's gone, I can talk to you about the stop and go signals of the cell cycle. Let's first talk about MPF and how that relates to the cell cycle. Cyclin is a protein that's accumulated in the late S phase and continues to accumulate in the G2 checkpoint. Once there's enough cyclin that's produced, it will activate the CDK, which stands for cyclin-dependent kinase. As it comes from the name, it's cyclin-dependent, so it'll only activate when it's interlocked with the cyclin. And now when this happens, it creates MPF, which is maturation promoting factor. But we can think of it as the M phase promoting factor because that is the go signal to go into the M phase and the stop signal for the G2 checkpoint. And so during N phase of the M phase, the cyclin is degraded and the CDK is recycled, and this could be a go signal for the G1 checkpoint and the stop signal for the M phase. Like I said, the CDK is recycled, so that'll come back to the G2 checkpoint and used when there's enough cyclin accumulated. Now let's talk about PDGF. PDGF stands for platelet-derived growth factor, but before we talk about that, let's talk about what growth factor is. So growth factor is a protein that's released to encourage cell division. So let's look at this experiment. In this experiment, human connective tissue is cut up into small pieces and placed into a petri dish. Then enzymes are used to digest the extracurricular matrix in the tissue pieces, resulting in suspension of free fibroblasts. Fibroblasts are just connective tissue cells. And now that is placed into two vessels, one without platelet-derived growth factor and one with platelet-derived growth factor. So platelet-derived growth factor is basically a specific kind of growth factor. It is made by blood fragments called platelets, and basically without the PDGF, the fibroblast fails to divide because it has nothing encouraging that cell division. But when there is PDGF, those fibroblasts are dividing and they're creating more because there is PDGF to encourage that cell division. So now, to relate this back to the cell cycle, fibroblasts have PDGF receptors on their plasma membrane. So when that PDGF is released, they can tell, and that is basically a go signal for the G1 checkpoint to go into the S phase to divide. So in an animal, let's say it gets a cut, PDGF is released, and those fibroblasts get that message, and the cells will divide and heal the wound. Now let's talk about disruption in the cell cycle. Cancer cells don't have the normal signals that regular cells do. They basically have a whole bunch of go signals. Everything is a go signal. There is no stop signals like the highly regulated cell cycle. They also don't have anchorage dependence or density dependent inhibition. Anchorage dependence is when the cell sticks to a substratum such as this dish surface, and once that cell is attached, then it starts growing inside from there. And that is what anchorage dependent basically is. So once it is attached to a dish surface, it will divide into a single layer and then stop dividing. And that is what's called density dependent inhibition. Let's say we were to scrape a part of that out. The cells will divide and only fill in that gap. And that is also what density dependent inhibition is. It'll know when to stop dividing. But in cancer cells, they don't have that.
In cancer cells, it'll continue to divide, forming a clump of overlapping cells.
This would be what we would call a tumor. A benign tumor is basically a harmless lump, and usually that can be removed through surgery. But a malignant tumor is a tumor that harms other organs, and if someone has a malignant tumor, you can say they have cancer. And now let's say that the cancer cells were to go through blood vessels and lymph vessels and travel to other areas of the body. That is called metastasis. When the cancer cells go from one point in the body to another, that is metastasis. So that's all to learn today, and thank you for watching the BioBrainies podcast. Bye.